And let's head on over to the studio now, where we'll sit down with Dr. David Gallegos from the U.S. Environmental Protection Agency. Thank you so much for joining us.
I'm happy to.
Let me just ask, what have been some advancements in single-cell profiling methods?
So since the advent of the profiling of the transcriptome of individual cells, which has been around for a few years now, there's been a veritable explosion in the number of other features of individual cells and primarily their nuclei that we are now able to profile. So these include things like chromatin accessibility, like DNA sequences, cell surface protein receptors, and there's been an even more recent and more exciting convergence of these into multimodal technologies that allow us to profile both the transcriptome and some of these features at the same time from the same cells, which allows just a higher resolution look at both each of these aspects as they pertain to rarer and less populated cell types, but more so the manner in which they interact with each other to influence how cells respond to toxicants and different stimuli.
And what insights have you been able to gain from all this work?
Well, so the key word here is resolution. In a lot of realms of toxicity, some of the forefront being developmental cells and neurons, for instance, there's a baked-in heterogeneity where there are tons of different cell types that aren't equally represented but can have much more profound effects than their population would suggest. So the ability to see how individual cell types that might not be the most numerous contribute to the mechanisms by which toxicants affect the response of cells is massive in resolving these very first components of adverse outcome pathways, being able to populate what's happening in all sorts of cells and all sorts of developmental lineages, and really understanding what's happening when cells are exposed to a specific toxicant.
Thank you so much for joining us. We appreciate having you.